Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. Now, when the DNA replication is happening or even the cell is actually being continuously being exposed to the different types of the mutagenic molecules or the different types of mutations happens within the cell, because when the cell is replicating, it has a proofread activity, it has utilizes that proofread activity to correct the sequences, but even then there are spontaneous mutations which are going to happen into the DNA, and these mutations are actually going to be detected by a well established machinery, and that's how it is actually going to participate and it's going to correct those mistakes. You know that if these mutations are not being corrected, they are actually going to lead to the accumulation of mutations, and ultimately it may actually cause the development of the different types of the cancers. So, DNA damage, as the name suggests, it could be occurs into a DNA due to the error into the DNA replications. It could happen because it caused the spontaneous lesions, or it could be because of the transposable DNA, or it could be because of the physical mutagens or the chemical mutagens. Physical mutagens, there are several examples of the physical mutagen, and there are several samples of mutagen which we are going to discuss. And then errors in the DNA replication is also very important in terms of the DNA damage. So, what is mean by the DNA damage? It is known that the genome is not a static entity, right? Genome is present inside the nucleus or inside the cell, right, in the case of prokaryotic system, and it is whenever you are actually getting exposed to a particular type of physical or chemical environment, it is actually getting exposed as well. So, and you know that the importance of the genome, right, it is actually hereditary material, so it actually going to follow the information to the next generation. So, it is important that the genome should be intact and it should not be get damaged. So, hence it is highly subjected to a variety of hereditable changes. A sudden change in the sequence of an organism genome that give rise to the alternate form of any gene is called as the mutations. As a result, the DNA gets damaged. These mutations are mostly recessive and lethal. Mutations are random, can occur anytime in any of the cell of an organism. It is not like that if the mutation occurs once, the same mutation cannot occur again. Mutation is recurrent, can cause again and again, right? The question comes, how does the DNA damages happens? On the molecular basis, DNA can be damaged in two ways. Some mutations may cause spontaneously, some mutations are induced by the mutagens. Spontaneous damage occurs without the treatment of the organism with an exogenous mutagens. It is mainly due to an error in replications, spontaneous lesions and the transposable elements. Whereas the induced mutations arises when the mutagen react with the parent DNA, which causes the structural alteration in base pairing. What is mean by the mutagens? Any agent which causes the frequency of the mutations is called as, which increases the frequency of mutation is called as the mutagens. Now, as far as the DNA damage is concerned, you can have the two main category. One is called as spontaneous damage, the other one is called as the induced mutations. So, within this category you have the multiple components, and these components are important for causing the DNA damage. So, agents causing the DNA damage, you have the agents which are causing the spontaneous mutations, and you also have the agents which are causing the induced mutations. Within the spontaneous mutations, you can have the errors in the DNA replications, you can have the tautomeric shifts, and spontaneous lesions. Within the spontaneous lesion, you are going to have the deaminations, depurinations and depyrimidations and the oxidative cleavage. Whereas the transpositions is another phenomena through which it is actually going to cause the spontaneous mutations. Whereas in the case of induced mutations, you are going to have the two different categories which can cause the induced mutations. One is called as the chemical mutagens, other one is called as the physical mutagens. Within the chemical mutagens, you are going to have the different types of agents which are going to cause the base analogs. So, you are going to have the chemical agents which are going to function as a base analogs, and that is how they are actually going to destroy the DNA. Then you can also have the de-animating agents. You are going to have alkylating agents. Most of the anti-cancer drugs or majority of the anti-cancer drugs are alkylating agents. And then you are also going to have the intercalating agents, because they all are actually going to do interfering into the replications and interfering into the repair mechanism, and that is how they are actually going to cause the mutations. And that mutation will perpetuate from the generation to generation. Then you are also going to have the physical mutagens. Physical mutagens means the physical parameters what you are going to use. So, in that case you are going to have the UV radiations, you are going to have the ionization radiations, and you are also going to have heat. So, these are the physical parameters which are actually going to cause the mutations. Now, let us first start with the spontaneous mutations, and then we will discuss about the induced mutations. So, spontaneous mutations, number one category is the error into the DNA replications. So, tautomeric shifts, right. For example, so shift of a proton from its nitrogenous base form its rare form, and that is called as the tautomeric shifts. The stable keto form of the thymine and adenine and the amino form of the adenine and cytosine undergoes tautomeric shift to form the unstable enols and imines respectively. For example, you have the stable form of the adenine and guanine, that is the amine form, and once it goes from the tautomeric shifts, it is actually going to form the immunoforms, and these immunoforms are very rare, but they are actually going to be less unstable. These forms are very short lifespans. If they are incorporated into nascent DNA, they may result into the mutations. If these bases are present in their unstable enol or the imino state, they tend to form the AC and GT base pairing. This AC and GT base pairing is not allowed, and that is how it is actually going to destroy it, or it is actually going to distort the DNA structures. And because of that, for example, this is C, it is actually going to form the interaction with the A form, right. So, A is always making a base pairing with T under the in the DNA, but in this case A is actually going to make a pair with C, and that is actually going to cause the alteration into the sequences, and that is how it is actually going to go into the next generation. So, imagine that you have a C. So, instead of if you have a C into the template, ideally it should be G into the replicated DNA, right. But instead, since this kind of mutation is happening, or this kind of tautomeric shift is happening, what will happen is that C is actually going to give rise to a synthesis of A. So, instead of G, you are going to have A into the DNA sequences, and that is how it is actually going to cause the mutations. The net effect of such event and the subsequent replication required to segregate and mismatch pair, that is the AT to GC or GC to AT base pairing substitutions, and that is how it is actually going to cause the mutations. Mutations via the tautomeric shift in the bases of DNA. In the examples, a guanine undergoes a tautomeric shift to a rare enol form at the time of replications. In its enol form, it is paired with the thymine. So, this is what exactly is what is showing here, that you are actually replicating a sequence where you have the all sort of nucleotides like G, C, T and all that, and during the replication, if the G is actually getting converted or tautomeric getting shift into enol form, then G is actually going to start making a pair with T. Instead of G is going to start making pair with C, and because it has altered the base pairing information, it is actually going to allow the formation of the, instead of GC, it is actually going to give you the AT, right, because it is actually going to give you the T instead of G, and as a result it is going to replicate. So, once the DNA replication is going to happen of this particular sequence, it is actually going to give you the, in the wild type it should be G is actually going to give you the C, but in this particular type of second generation, instead of G and C, it is actually going to AT, right. So, this is what exactly happened when you are going to have the tautomeric shifts. So, during the next generation, the guanine shift back to its more stable keto form. the thymine incorporated opposite the enol form of the guanine direct the incorporation of the adenine during the subsequent replication. The net result is that the GC is going to be 80 mutations. So, G is making a base pairing with T. So, as a result in the first generation the T is going to be synthesized, and then in the second generation G is again reverting back into the keto form. So, because of that it is actually going to start again synthesizing the G to C, but here instead of T it is actually going to, since you have the T in the daughter, it is actually going to start synthesizing the A, and as a result what will happen is that wherever you having the G, it is going to be replaced by A. So, it is actually going to have the G to A mutations, right. And if you have G to A mutations, then it is actually going to change many things. It is going to change the amino acid, what is corresponding to the G to A substitution and so on. And then you have the substitution mutations. So, error during replication results to the substitution mutation or the Frameshift mutations. So, substitution of one base pair into another is called as a substitution mutation. The swapping of the base pair may be a transition mutation or the transversion mutations. So, what is the transition mutations? Substitution of one pyrimidine by another pyrimidine or one purine by another purine is called as the transition mutations. For example, G to C is going to be replaced by A and T and vice versa. So, in that case it is actually going to cause the substitution mutations. Then you have the transversion mutations. So, purine is replaced by the pyrimidine or the pyrimidine is replaced by the purine. So, subsequent transversion is transition, for example, G to A or A to G or C to T or T to C. These are the, if the G is replaced by A, then it is going to be called as transition mutations, or A is replaced by G, then it is also going to be called, because the pyrimidine is replaced by the pyrimidine and purine is replaced by the purine. But if it is the pyrimidine which is replaced by the purine, then it is going to be called as transversion. So, both of these kind of substitutions are actually going to be very problematic, because it is going to overall change the amino acids and it is going to cause the mutations into the subsequent gene products. Then you have the Frameshift mutations, right. So, sometime it may happen that during replication some extra nucleotide may get inserted or may not get copied, right. So, you can actually have the either the addition of the extra nucleotides, or you can actually have the disappearance or the deletion of some of the mutations, and in both of these cases it is actually going to cause the Frameshift mutation, right. This frame information anyway you will be able to understand when we are going to discuss about the translation, because you know that the protein is going to be synthesized in the form of the codons, right. So, and codon is made up of the 3 nucleotides. So, protein is being synthesized in the form of the codon, right. So, for example, this is the codon, right, ATG. So, these 3 nucleotides are going to be read together by the anticodon what is present onto the tRNA. But now imagine that if I add one more A into this, so what will happen is it is going to be this sequence, right. Now, if I go by the 3 triplicates, then it is actually going to be like this. This means earlier the codon was ATG, now the codon is going to be ATA, and codon for ATG is going to be X amino acid, whereas codon for ATA could be Y amino acid, and that is how there could be a change in the amino acid what is going to be incorporated into the protein. The another example is that, for example, if I have the A here, right, and if I or C, for example, right. So, if I have a sequence like A, T, G, C, A, right, and if I remove, actually if I remove, for example, if I remove this actually, right. So, if there will be a deletion, then what will happen is it is going to have this. So, earlier you were having this as a codon, now you are going to have this as a codon. So, this also is going to be cause the deletion. So, this is going to happen because of the deletion, this is going to happen because of the incorporations. So, if this happen in the exonic region of DNA, that may mainly change the translational reading frame, hence results in the production of a non-functional protein which can be observed in the phenotypic characteristics. So, it is not only going to change the codon for this, it is actually going to change the codon for subsequent generation also. For example, it is going to change the frameshift mutation. So, it is actually going to change the codon. For example, earlier you were having this. So, this is the first codon, this is the second codon. Now, if you remove this T, right, then you are going to have the first codon as AGC and the second codon would be ATT, whereas earlier it was ATG and CAT, right, CAT. So, this was the codon, right. So, this is called not only going to change the frame shift only for first codon, but also for the all the codons, and because of that it is actually going to cause a significant change into the amino acid composition of the product, and as a result it is actually going to produce the protein which may not be functional and which may actually cause the problem to the cell. For example, this is the example what is being given, right. You have, this is the standard for frames what is present, and when you have the mutations or the messenger RNA what is going to be formed, it is actually going to form. But if you have the addition of T base pair, then it is actually going to change, right. It is going to change the frames, and that is how it is actually going to form the proteins. Or if suppose you have the deletion, then also you are going to change the things. So, these are the some of the things which are very important, and the frameshift mutation is very significant problems. And as a result, this shift to change in the translational reading frame, hence name as the frameshift mutations. Then we have the second thing is spontaneous lesions, right. Naturally occurring damage to the DNA is called as the spontaneous lesions. Most common lesions are deaminations and depurinations. So, deaminations, the loss of exonucleic amino acid group from the cytosine and adenine and the guanine due to change in the pH in temperature, is spontaneously resulting in the formation of uracil, hypoxanthine, xanthine and thymine respectively. So, deamination is going to change the amino acid. For example, if there will be a deamination from of these nucleotides, then it is actually going to be get converted into uracil, hypoxanthine, xanthine and thymine, and that is actually going to result into the change in the structure or change in the nucleotide sequences. Then depurinations and depyrimidations, loss of purines and pyrimidine by the breakdown of the glycosidic bond nucleotide from the DNA due to damage due to the change in the pH. And then you also going to have the oxidative damage, damage in the DNA due to the reactive species spontaneously, radicals like peroxide, hydrogen peroxide and hydroxy radicals attacks on the DNA product produce variety of products. Attack at sugar residue leads to the fragments, base loss and the strand streak. So, all of these things can also lead to the frameshift mutations and the spontaneous mutations as well. So, these are the some of the example. If you have the deaminations, the cytosine is going to be get converted into uracil. If there will be a deamination into the adenine, it is actually going to be get converted into the hypoxanthine. And then if you have the 5-methyl cytosine, and if there will be a deamination, it is going to be get converted into the thymine. And if there will be a deamination into the guanine, then it is actually going to be get converted into xanthine, and some of these are non-natural amino nucleotides. So, they will actually going to cause the significant mutations into the DNA structures. And then we have the transpositions. Transposon is a DNA segment that have the capacity to insert itself at any location in the genome without having any relation with the target sequence. Consequently, it causes loss of gene function or genes inappropriate overexpressions. So, transposition is a very important and a very significant topic that you should actually study, and transposons are also called as the jumping genes, and they are actually changing its position from the one locus of the genome to the another locus, and as a result they are actually causing the different types of artifacts and different types of problem into the genes. So, they are actually going to cause the gene loss of gene functions or genes inappropriate overexpressions. So, transposon is a very important and a big topic, so that you can actually be, if you are interested, you can be able to study this from any of the standard another molecular biology books. And then we have the induced mutations, right. So, mutagens results into the induced mutations. Mutagens can be classified as the physical mutagens and the chemical mutagens. So, these are the physical mutagens where you have the UV radiations, ionizing radiations and the heat. The UV radiation is a potent physical agent that cause a number of photoproduct in the DNA. UV radiation of the wavelength 260 nanometer induced dimerization of the pyrimidine nucleotides bases, especially thymine, resulting in the formation of the cyclobutyl dimers. So, this is what exactly happened, right? When you have the two nucleotide, they will be getting exposed to the UV, they are actually going to get dimerized and they are actually going to form the cyclobutane rings. Adjacent pyrimidines are covalently linked to the formation of 4-membered ring structure, and this structure is called as the pyrimidine dimers. Then ionizing radiation mainly causes the DNA strand breakage, and then you also have the heat stimulate the wave induced cleavage of the N-glycosidic bond, which results in the apyrinic or apyrimidinic side or the baseless sides. So, in this particular thing, what will happen is that there will be a cleavage of the glycosidic bond, and you know that the glycosidic bond is holding the sugar to the base, and because of that there will be no base what is present onto these particular nucleotides. Then we have the chemical mutagens. So, in the chemical mutagens you can have the 4 categories: the base analogues, deaniminating agents, alkylating agents and the intercalating agents. Base analogues are certain bases which are not present in the DNA normally, but resembles to the normal nitrogenous bases that can incorporate during the DNA synthesis. For example, the 5-bromouracil, which was base analogues to the thymine, and the 2-aminopurine, which is analog of the adenine. So, these are the base analogues are going to induce the mutations because they are going to mimic the natural bases, but they are not the natural, and they will be get incorporated into the DNA during the DNA synthesis. Then we have the deanimating agents, which causes the point mutation by removal of the amino group from the nitrogenous bases. Nitrous acid deaminate the adenine, cytosine and guanine. Sodium bisulfite deaminate only cytosine, and the deamination of adenine give rise to the hypoxanthine, which pair with C instead of T, and deamination of cytosine give rise to uracil, which pairs with A instead of G, and this actually is going to cause the problem into the first generation and as well as into the subsequent generation because of the C to T mutations and A to G mutations. Then we have the alkylating agents. So, some example of alkylating agents are like ethylene methane sulfone, or the nitrogen mustard and the dimethyl nitrosamine. These agents actually add the alkyl group to the certain position in the nucleotide, and these alkylating agents are actually going to cause the mutation, and that's how it's actually going to kill the cells. Many of the anti-cancer drugs are also alkylating agents. So, they alkylate the DNA, and that's how they are actually activating the machinery to to kill these cells. And then we have intercalating agents, usually associated with the simple single mutation, pair insertion or deletions. Intercalating agents are flat molecules that flip between the base pair in the double helix, resulting into the unbinding of the DNA helix and therefore increasing distance between the adjacent base pairs. Some examples are proflavin, acridine orange, ethidium bromide and the ICR compounds. Intercalating agent is also very problematic because they are going to interfere into the DNA synthesis, and they are also going to change the normal DNA structures, and that's how they are actually going to cause the mutations. So, these are the one of the effects. So, for example, if this is the DNA, right, how the base analog is actually causing the problem, right. So, for example, this is the normal DNA and you have the A, which is, and in the corresponding template you are going to have T. Now, if there will be a base mutation, so for example, the 5-bromouracil is going to be present instead of A. So, if there will be a BU undergoes tautomeric shift, then BU is actually going to get converted into this, and that's how it is actually going to have the affinity for the C rather than G, because this is going to have the affinity for G rather than C, but rather than A, because AU should have an affinity for A, but it does not have the affinity for A, it has affinity for G. So what will happen is, when the replication occurs in the wild type, the A is going to form and A is going to be synthesized in front of T, but in this one, instead of this, you are going to have the G. Now once you do the another round of applications, this wild type will not actually have any problem, because it's going to still have the A to T, but here now the G is first mutated, and now the second strand is also going to be mutated, and that's how it is actually going to have the C. So this is actually going to be a mutated DNA what is going to be formed, and this mutated DNA, if you do the subsequent replications, will proliferate, and it will continue into the subsequent cells actually, or neutrototal cells. Similarly, you can have the deaminating agents. For example, adenine, when it's going to be attached to the nitrous acid, it's going to get converted into hypoxanthine, or the cytosine, when it's going to get converted into AT. So once you have the adenine to the hypoxanthine will not going to form the interaction with T. Instead, it is actually going to have the interaction with C, and because of this, the AT is actually going to be replaced by GC, exactly with the same mechanism that in the first generation the A will actually going to recognize the T, but in the subsequent generation, when the its A will get converted as hypoxanthine, the daughter strand is going to have the cytosine, and cytosine is actually going to incorporate the C. So that is what exactly going to happen in the subsequent generation, even with the deaminating agents. And then you also going to have the alkylating agent. For example, guanine is going to get converted into the ethylene guanine, and ethylene guanine is going to have the affinity for thymine instead of the interaction for cytosine, and because of that there will be a reverse, right. In this case, the AT is being replaced by GC. In this case, the GC is going to replace by AT, and the same is true for the thymine. When it is getting alkylated, it is going to form the ethylene thymine, and ethylene thymine have an interaction or going to make the base pairing with the guanine rather than the adenine, and as a result the TA is going to replace by the CG. So these are the some of the mechanism through which the DNA is going to be mutated. Now how the DNA, if the DNA got damaged, right, whether it is going to be damaged by the spontaneous mutations or it is going to be damaged by the induced mutations, you are supposed to repair these changes, right. You are supposed to have the machinery to detect, and then if possible you can actually be able to have the repairing of these things. So systems for repairing the numerous unintended lesions that frequently occurs in DNA are also required for maintaining the genetic stability. In addition to the extremely precise DNA replication mechanisms, the vast majority of these unintentional DNA replications are temporary, because the DNA repair a group of connected system immediately correct them. Without repair processes, a genome cannot maintain its essential biological functions. These DNA repair mechanisms fall into the two broad categories: direct reversal of the chemical process that caused the DNA damage, and the damaged bases are removed and then replaced with the freshly synthesized DNA. So you have the two choices of reversing these damages. First, you can actually be able to reverse the process. You can actually reverse the reaction. For example, if you have the alkylating agents, you can actually reverse the alkylating agent and reverse these reactions, so that the guanine is going to be converted into back to the adenine and so on. Or other thing is that you can just replace this damaged base and replace it with the normal base, and that is also going to be another base. So additional mechanisms have developed to help the cell deal with the damage where the DNA repair fails. DNA repair mechanisms, you can actually have the single strand break repair mechanism or the double stranded break mechanisms. In the single strand break mechanism, you can have the direct reversal, excisions, mismatch repairs, and within the excision repairs you can have the nucleotide excision repairs or the base excision repairs. Whereas in the double strand breaks, you can have the homologous recombinations or the non homologous DNA recombinations. So single strand breaks, you can have the direct reversal. So, directly acting on the damaged nucleotide, converting back to the original structures. So, pyrimidine dimers are usually repaired by a light dependent direct reversal process called as the photoreactivations. DNA photolyase enzyme participate in the mechanism, which is found into the E. coli. These enzymes get activated with the wavelength of the 300 nanometer and the 500 nanometer. The enzyme binds to the pyrimidine dimer and convert back to the original monomeric and nucleotides. So, this is what exactly happened when you have the thymine dimers which are going to be formed, and when you are exposing this particular DNA with a visible light, it is actually going to have the photolysis of this particular strands, where you are actually having the nucleotide dimer which is being formed, and then with the help of the enzyme which is called as DNA photolysase, it is actually going to replace and it's going to remove these nucleotides and it's going to form the normal DNA. So, these are going to reverse, and that's how it is actually going to recover the DNA. Then we also have the excision repairs. So, the excision repairs, it involves the excision of the damaged segment of DNA followed by the resynthesis of the current nucleotide sequence by an enzyme which is called as DNA polymerase. So, base excision repairs, you can actually have the removal of the damaged base used to repair minor damages like the alkylation, deamination, which are a consequence of exposure to the mutagens. It is initiated by an enzyme which is called as DNA glycosylase, and the DNA glycosylase cleaved at glycosidic linkage, hence detaching the altered base. As a result, an apurinic or apyrimidinic sites are generated. Final ligation of the nucleotide takes place by the DNA polymerase and the DNA ligase. So, this is the mechanism where you are actually having the UDG role of the UDG, uracil DNA glycosylase. So, if you have the deaminated cytosine which is actually going to be uracil, and that is actually going to first form the apurinic sites, and then these apurinic sites are actually going to refilled back, and that's how you are going to have the repaired DNA at the end of this particular repairing mechanisms. Then we can also have the nucleotide excision repair. So, similar to base excision repair, it acts on a more substantially damaged area on the DNA. It includes the following steps. So, damaged DNA, first you are going to have the damaged DNA, the breakage of the phosphodiester bond on the either side of the damaged portion. So, for example, this is the damaged DNA. So, on the both side you are going to have the breakage of the phosphodiester bond, and then you are going to have the excision leaves gap, right. So, you are going to have a gap which is going to be created, and this gap is going to be filled by the DNA polymerase and ligase seal the breaks. The best studied mechanism for the DNA repair is the UVR system into the E. coli. A key enzyme is involved called ABC exonuclease, which is made up of the 3 subunit UVRA, UVRB and UVRC genes, and ABC exonuclease binds to the damaged site on the DNA and cut the phosphodiester bond on the both 5' and 3' lesion. UVRD is acting as a helicase and helps to unwind the DNA at the site of the cut. The gap is filled by the DNA polymerase 1 and sealed by the ligase. And then we have the third mechanism. The third mechanism is called as the mismatch repair. It detects the mismatch that occurs during the DNA replications. The mismatch repair occurs in daughter strand, and it highly prone to the mismatch during the replications. How to distinguish between the parent strand and the daughter strands? Immediately after the replication, parent strand will be containing methyl group, whereas the daughter strand await for the introduction of the methyl group. In this way, the two strands can be distinguished, and this is the best time period for the repair mechanism to scan the lesion like the correction. So, this is very important that see, the damage will only going to occur into the daughter cells rather than the parent cell. So, parent cell you are just going to remain as intact. In the daughter cells, you are going to have these kind of repair mechanisms. And how you are going to recognize the daughter cell? The daughter DNA is going to be unmethylated, because the methylation still not been done for all the adenine groups. How the methylation occurs? In E. coli, the methylation is executed by an enzyme which is called as the DNA adenine methylase, which converts the adenosine to the 6-methyladenosine in the sequences like GATC, and the DNA cytosine methylase, which converts the cytosine into the 5-methyl cytosine in the 5' this sequences. It should be noted that these methylations are not mutagenic. The modified version have the same base property as the unmodified one. And then E. coli, the mechanism is completed by the mut protein. There is the involvement of 3 mut proteins, the mut H, mut L and mut S. Mut H and mut L recognize the sequence GATC and mismatch respectively. And then we also have the SOS response. So sometime a DNA damage is so dangerous that it stimulates the cells to produce the DNA repair enzyme that allows an immediate reaction to the specific DNA damage. SOS response in bacteria is the most researched illustrations. When the bacterial chromosome is severely damaged, the SOS response genes are activated. Many of these genes are involved in a DNA repair and mutagenesis. Rec protein and LEXA repressors are the main player in the SOS response. LEXA is a repressor that binds to the 20 base pair segment of DNA known as SOS box to prevent the activation of the SOS response genes. As a result, the LEXA govern the transcription of the each SOS response genes. So this is just a mechanism through which the SOS response is happening. So you are going to have the SOS box where the LEXA gene is going to bind, and that's how it is going to start the SOS response gene transcription. And there will be no transcription of the SOS response, so there will be no damage into the chromosomal DNA. But if there will be a damage into the chromosomal DNA, then the LEC A, you are going to have the recruitment of REC A, and that's how it is actually going to cause the cleavage of the LEXA. And once there will be a cleavage of LEXA, then this inhibition or the attenuation is going to be removed, and that's how the SOS box is free, and that's how it is actually going to cause the production of the SOS response gene, and that's actually going to cause the bacterial chromosome damage repairing. And then we also have the double strand breaks. So far what we were discussing about the single stranded break repair mechanisms. In the double stranded break mechanisms, you have the two mechanisms, the homologous recombinations and the other kinds of mechanisms. So these are the some of the steps what you are supposed to follow when you are going to have the homologous recombinations, and these steps are very important, and we are going to discuss in detail about all these steps when we are going to discuss about the genome editing and the other kinds of phenomena in a subsequent lectures. Then we are going to have the non-homologous DNA repair and end joining, and all these we are actually going to discuss, because the non-homologous process repair double stranded breaks in the DNA, and this process is known as homologous because of the break DNA are directly ligated without any homologous template. The term was coined by the Mori and Haber in the year of 1996. And double stranded breaks are recognized by a protein which is called as Ku70/80, and the Ku70/80 recruit the DNA PKs or the kinases, and it then recruit the Artemis, which remove the damaged ends. So in conclusions, DNA damage is inevitable due to the various factors. Cells have evolved repair mechanism to counteract the DNA damage. Types of DNA damage includes the breaks, base modification and cross links. Repair pathway like the BUR, NUR, MMR and HR fix the different type of DNA damages. Accumulated unrepaired DNA damage can lead to the mutation and disease, including the cancers. Defective repair mechanism contribute to the cancer development. Some cancer therapies exploit the DNA repair mechanisms, and that's how they can actually cause the mutations into the cancer cells. And since you are going to have several mutations, the cancer cell will have no option but to die actually. Aging is linked to the declining DNA repair efficiency. The environmental factor can overwhelm the DNA repair mechanisms, and ongoing research are seek to improve the understanding and develop the therapies. So there are many mechanisms. Once the DNA is damaged, there will be many mechanisms through which the DNA is being repaired into the eukaryotic cell or the prokaryotic cell. So this is all about the DNA damage and repair mechanisms, and we have discussed various mutagenic molecules which are actually causing the induced mutations. We have also discussed about the spontaneous mutations and how the spontaneous mutation is happening because of the error prone, because of the DNA replications and the other phenomena. So with this I would like to conclude my lecture here. Thank you.